All right, everybody, let's get started. Good afternoon, folks. Welcome to the North Carolina Museum of Natural Sciences into the SECU Daily Planet Theater. This is the Lunchtime Discovery Series. It's an opportunity we take every Wednesday to meet interesting people who are doing interesting work out there in the worlds of science, nature, conservation, education, and even more. And oftentimes we're talking about those things as they're happening here in the state of North Carolina. The Lunchtime Discovery Series is coordinated and organized by the folks at the Office of Environmental Education within the Department of Environmental Quality. Many thanks to them for helping to pull this together to bring the speakers into the museum. And then we broadcast it and host it here at the Museum of Natural Sciences, because really, what better place to talk science and nature in North Carolina than at its biggest natural history museum, and some might argue, but I think the best natural history museum, the best place to do it. So, for today's program, really cool stuff. Here at the museum, if you're like a visitor walking around our museum, you cannot miss the 7 different whale skeletons that we have on display. Our museum is well known for our fantastic whales, and we think about whales a lot here at the Museum of Natural Sciences. And so I'm very excited for today's guest, who's going to give us some interesting and I think very cool insight into what's happening with some very interesting, and as I've learned, enigmatic species of whales too. So very excited that today we have the director of the Duke Marine Lab, Dr. Andy Reed, to talk about some research that's going on just recently. Everyone, please put your hands together and welcome Dr. Reed to the stage.
Thank you so much. I really appreciate the invitation to be here and to tell you some about these crazy enigmatic whales. I love museums. I got my start working on whales at the Ontario Science Centre in Toronto, working on a fin whale skeleton many years ago. And I just love the ethos of museums. I love the research and conservation that they do. So I'm ecstatic to be here. I do want to put a little plug out for a local museum down on the coast in Beaufort where the Duke Marine Lab is, and that's the Bonehenge Whale Center. I think they might be online right now, operated by Keith Rittmaster. And it's a museum, small museum and center that's dedicated to articulating whale skeletons. And we have a great example of a goose-beaked whale at the Marine Lab that Keith and his team put together for us. They do beautiful work, so if you're in the area, get online, look them up, and arrange a visit. It's a wonderful resource for the state and for the coast. Okay, I'm going to talk today about beaked whales, and you probably don't know much about beaked whales because they're so poorly understood. There are something like 80 or 90 species of cetaceans, whales, dolphins, and porpoises, depending on how you count them. And one of the most diverse groups of cetaceans are the beaked whales. But because they're deep diving animals and because they typically live very far from shore, we know very little about them. And if I was giving this talk 20, 25 years ago, I would be telling you about what we know from stranded animals because nobody was really studying them in the wild. That changed in about 25 years ago when we had a mass stranding of Cuvier's beaked whales, as they were known then. Now we refer to them as goose-beaked whales, and other species in the Bahamas. And it's unusual for beaked whales to strand en masse in multiple, in large numbers. Concurrent with that stranding was a Navy exercise in which U.S. Navy vessels were conducting anti-submarine hunting exercises. And a report that NOAA and the Navy put together to try and understand what had happened concluded that the sonars that the Navy vessels had been using were most likely responsible for the strandings of these whales. And this followed on some earlier episodes that had happened in places like the Mediterranean and the Canary Islands, in which similar strandings had occurred. And it brought a lot of attention and concern about the effects of these sonars on these deep diving whales that were really poorly at that time understood. There was a lot of consternation in the Navy that their training exercises might be harming marine wildlife. And so they funded a considerable amount of early research to try and understand what had happened. Was it acoustic trauma? Was it the intensity of the sound? Was that something in the signal that the animals were interpreting in a strange way? And the hypothesis arose that beaked whales might be interpreting the sonars that were being used in these exercises in a similar way as they would interpret the sounds of predators. And if I were to play for you the sounds of some of these tactical sonars, they sound to our ears a lot like the sounds produced by killer whales. And killer whales are the primary predator of beaked whales. So that was kind of the hypothesis that people started to think about, trying to understand this phenomenon. At that time, we had no way of really studying these animals. And so, again, with funding from the Navy, two researchers, Peter Tyack, who's on the left there, and Mark Johnson on the right, developed a new tag, a digital acoustic tag that could be applied to the backs of these whales with suction cups. And if you're here in the museum or you come and visit the museum, there's a great display out near Stumpy talking about these tags. So these tags we applied them for short periods of time for about a day or so. They record the sound that the animal produces, the sound that the animal hears. They have accelerometers, they have a depth sensor, and eventually they detach from the animal, float to the surface, we retrieve them, and download the data. I always tell my students it's kind of like having your iPhone on the back of a whale for a day, so you can find out what it's doing. This allowed researchers for the first time in the Bahamas to tag one of these whales and to play back simulated sonar signals to them. And so this is from about a little bit more than 10 years ago now. Researchers were able to, for the first time, actually document the response of these deep diving whales to these tactical sonars. And they did respond in a way that was consistent with the way we would expect them to respond to predators. When they heard the sound of the sonar, the beaked whales stopped producing any sounds themselves. They went silent and they moved away from the source. So just a classic kind of cryptic response to the presence of a predator. You try and hide and you move away and you stay quiet. I won't go through all the data, but if you look at that top plot, it's just an example of depth. You can see the animals diving, making kind of what we refer to as bounce dives, and then very long foraging dives. And in the blue there, that's when the animal's actually producing, the beaked whale's producing sound to hunt prey. The red is the exposure to the sonar, and you can see that the animal immediately goes quiet and then stops its foraging dive, goes to the surface, and then makes a whole series of relatively shallow dives and moves away from the source. About the same time as that study was conducted, we began studying pelagic whales off Cape Hatteras. So Cape Hatteras is an enormously rich area, as you likely know, because it's the place where the warm waters of the Gulf Stream, bringing subtropical species, collide with the cold waters of the Labrador Current, bringing temperate and boreal species from the north. So we work off the edge of the shelf off Cape Hatteras. We run in and out of Oregon Inlet. It's about a 40-mile ride to our study site from Oregon Inlet. We first started becoming interested in beaked whales there when one of my PhD students, Joyce Tanistreet, deployed acoustic recorders to understand the distribution of beaked whales along the US East Coast. So these are the places that she deployed her recorders, one off Nova Scotia, one off Georges Bank, one on Norfolk Canyon, Hatteras, Onslow Bay, and then down off Jacksonville, Florida. And these are bottom-mounted recorders that sit and listen for a year, and they record all the sounds they hear. And when Joy did the analysis of the recorder off Hatteras, she found that goose-beaked whales were found almost every day there. So it was our first hint that this species of concern to the Navy was found in our backyard with a frequency that might be possible to allow us to study them. Shortly after, our colleagues at UNC Wilmington, Ann Pabst and Bill McClellan and their team conducted aerial surveys for the Navy in this area. And you can see the transect lines there that the plane flew. And these are sightings of goose-beaked whales in the blue squares, and another species of beaked whale, Mesoplodon europaeus, Gervais beaked whale, in the red triangles. And then the green squares are likely Gervais. But you can see off Cape Hatteras, if you go just kind of north and east of Cape Hatteras, there are a lot of blue squares there. And what Ann and Bill's team found was that the animals were found year-round and found in very high density. Interestingly, as you come around the Cape, the density of goose-beaked whales drops off pretty significantly, and those animals are replaced largely by Gervais beaked whales. We really don't understand why that is. At the time, we weren't working on beaked whales. We were working on short-fin pilot whales, the species here in front of our research boat. We were interested in the biology of these animals. We were interested in their interactions with a fishery out there, a pelagic longline fishery, because these animals have learned to take captured fish and bait off these long lines. And so we were trying to sort that out. But every now and then, members of our team, including the husband of somebody in the audience here, would wander off and go out into the really deep water. And what they found when they went out there were goose-beaked whales. And this is a picture. It's about another 10 miles off the shelf break. This is a classic group of goose-beaked whales. There's an adult male in the foreground, and then either juveniles or females, those browner animals, in the background. So we started working on these really difficult to study animals. And over the last 10 years, we've kind of perfected our techniques, allowing us to apply a variety of different types of tags. And the reason that we've been able to do this is because we have an extraordinarily skilled field team. And I just want to mention them by name because it's a privilege to be able to work with people who are so skilled. So going from the top to the bottom, that's Zach Swaim, who runs our field operations. Daniel Webster, who's a wildlife marks person, so he actually uses an air gun to fire implantable tags into the dorsal fins of these animals at sea, which is a remarkably difficult thing to do safely. Heather Foley, who did a lot of analysis of the data that came in, has moved on since to go work for NOAA, working on right whales. Will Chofie, who did a PhD in my lab, and then Annie Harshberger at the bottom there, who's currently doing a PhD in my lab. And these guys are extraordinary, as I said, in the field, and allow us to study these really, really enigmatic animals. So I'm going to give you just a little taste of what it's like out there. We select very calm days because the animals are difficult to see, they're very shy, they're very difficult to approach, so we need conditions to be pretty flat. So we spend a lot of time looking at weather models, trying to find days that it's going to be nice, won't be too windy, and the ocean will be flat. So I'll just give you a sense of what it's like. Here we are 40 miles off Cape Hatteras. We've seen a group of beaked whales, and we're closing in on them rapidly because typically they only spend about 2 minutes of time at the surface before they submerge again. So you're looking at a GoPro on Will Chofie's camera. There's Zach driving. And you'll see Will's about to go up into the pulpit, and you'll get a sense of what it's like when we approach one of these groups of whales. Time is of the essence because the animals spend so little time at the surface. So in that 2 minutes, we have to close to the group without spooking them. We have to be able to get close enough so that we can take pictures, and if we want to deploy tags, to be able to do that before they submerge. So here we have a group of 4 animals. You'll see we get closer. There's one adult male in the group. And interestingly, the adult males often interpose themselves between us and the females in the group. And so we actually have a bias when it comes to tagging these animals because we're almost always tagging adult males. They're also very distinctive and easy for us to identify. And the females and the juveniles, the brown animals, are much, much harder for us to identify. There's a female, the adult male's right in front. You can see conditions are really nice. When we have days like this, the 2-hour commute out to our study site is worth it. Sometimes we get out there and it's not quite so nice. You'll see just in a second, Will's about to turn on the lasers on the camera. Those lasers are a known distance apart, so if we get a picture of the animal with the 2 laser dots on the side of the animal, we can actually estimate how big the animal is. Okay. Now let's talk about applying these tags. This is applying digital acoustic tags. So these tags are applied using a carbon fiber pole. That's Zach up in the pulpit. I'm driving. He is telling me that I'm going right by a beaked whale, which I am. I didn't mean to do that, but that's what happens. But fortunately, there was another animal that Zach can tag. So you take the pole, you slap a tag on, 4 suction cups. And those suction cups we can program to release at a predetermined time. So typically we keep those tags on for about a day. And again, for that day, we have a complete record of where the animal was in the water column, the sounds it produced, the accelerometry it experienced, and the sounds it hears as well. The other type of tag we use are the longer-term tags that Daniel shoots from the air gun. And this technique was developed by this group of researchers. And so we learned from them. They were deploying these tags in California. And we've used exactly the same technique to tag animals off Cape Hatteras. So these tags are quite simple and straightforward. They just record pressure, which allows us to estimate depth at predetermined intervals. And when the animal comes to the surface, they dump that data up to orbiting satellites. And the satellites can use the Doppler shift, the perceived variation in frequency of this signal transmission to estimate where the animals are. Because the animals spend so little time at the surface, it's very difficult to be able to transmit very much information. And the position estimates we get aren't very good, and we have to continually to try and get the tag to send up data to make sure we have complete data records. I'll talk a bit more about that in a second. So the first thing we were interested in is, are these resident animals, do they live in Cape Hatteras? Do they move between New England and Florida? We knew nothing about them. And it turns out, this is work done by Heather Foley, who, while she was in my lab, did her master's at NC State, right next door here. By looking at the tracks of those animals that were tagged with the satellite-linked recorders, it's very clear we have a resident population of Cape Hatteras. The animals will sometimes go walk about for a little bit, wander around for a little bit, but they return to this area. We don't know what makes this area so special because the animals are foraging down at the bottom, as I'll show you in a second, at depths of about 1,500 meters. And we just don't know what's happening down there. We spend a lot of time wondering what is happening down there, but there's something about the confluence of the Gulf Stream, the edge of the shelf, that provides enough nutrition for these animals in the form of deepwater squid, deepwater fish. Many of those species, the prey species, are very poorly known. So this is an example of the diving behavior of these whales. These animals are extraordinary divers, the consummate mammalian diver. So this is work done by Jeannie Shearer, another PhD student in my lab, and this is a typical example of the dive profile. So depth is on the y-axis here, time is on the x-axis, and you can see that the animal repeats this kind of same pattern of a very long dive, which we interpret to be a foraging dive. The animals are foraging, going down very close to the bottom, interspersed with a series of what we refer to as bounce dives, which we'll talk about the function of those. But in those deep dives, that's when the animal's foraging. So my friend Dan Paps likes to remind students when she's talking about these animals, imagine that you go for a 3-kilometer jog. You take a breath at the beginning of your jog, and then you hold your breath for the next 3 kilometers, for the next hour or so, and you take the next breath when you get back to the surface. It's remarkable. So we think that this kind of behavior has evolved because the animals forage down near the bottom on relatively slow-moving prey, so it's a good place to go and feed, but they have to make this commute between the surface where they must come to breathe and to the bottom where they feed. And kind of paradoxically to us, the surface is a dangerous place for beaked whales because that's where killer whales are. That's where they are at risk of predation. They seem to be much more at home at depth because their killer whales are poor divers and can't get to those depths. So there's this really kind of interesting flip of our perception where the deep sea to us is strange and frightening. To the beaked whales, the surface is a strange and frightening place and the deep waters are safe. So this is just an expanded example of that. Again, just showing you this kind of really typical, stereotypical behavior. We refer to the time between those deep foraging dives as the interdeep dive interval. So we call those short dives bounce dives. I don't need to go into the details here, but we can easily separate the deep dives from the foraging dives from the bounce dives using the duration and depth. So on average, the animals are diving to 1,500 meters. So that's a round trip of 3 kilometers. As I said, they're going down and they're going to forage, and those dives last for about an hour. So imagine when we come to a group of animals, if they're going on a foraging dive, when we find them, we have to stay at the surface and wait for them to come back for an hour before they reappear. And when they're at the surface, they spend very little time there, only about 2 minutes, as I said. And we think that that is to minimize their risk. Interestingly, when they're at the surface and when they're making those shallow dives, the animals are silent. They don't make any sounds at all. When they're going on one of the long foraging dives, when they get to about 300 or 400 or 500 meters, they start to echolocate, they start to click. So they use a biosonar to find prey. So just imagine those animals down 1,500 meters in the canyons, searching for large, slow-moving fish, using a sonar when it's completely dark, it's 4 degrees Celsius, there's immense pressure. How does a mammal do that? We really don't know. So with that baseline, we felt we had enough information about this population of goose-beaked whales to pitch to the Navy the idea that we could actually conduct an experiment to see how the animals responded to tactical sonars. The studies that had been done to address this question before had all taken place on Navy ranges, the Bahamas and Southern California, and we wanted to be able to conduct an experiment in a place where sonar was not used very frequently. This is, I think, the most complicated experiment that I've ever been part of. It's a multi-institution, multi-individual initiative that's led by my friend and colleague, Brandon Southall. But there are an enormous number of people, including many people at the Navy, who make this work, and I just want to say thank you to all of them. I'll thank them again at the end, but this is very, very complicated. That's our research vessel in the background, the Shearwater from the Duke Marine Lab, and that's a goose-beaked whale in the foreground wearing one of our digital acoustic tags. So our objectives of this project, which we refer to as the Atlantic Behavioral Response Study, are to understand how these enigmatic whales respond to tactical sonars in relation to how loud those sonars are perceived by the whale, what the behavioral state of the whale is, if it's foraging or if it's in one of those interforaging intervals, and a whole bunch of other contextual factors that turn out to be quite important. And we wanted to, as I said, to conduct the study in a place where sonar was not used very often. Most of the anti-submarine warfare exercises on the East Coast occur in Norfolk Canyon further north. And we also wanted to extend the period of time in which we were monitoring animals. All the studies prior to this had just used digital acoustic tags, so we only had a 24-hour window. And we wondered, what happens after that 24 hours? Are animals still disturbed? Do they move away? Do they stay away? And so we wanted to use the satellite link tags to be able to understand that better. The way we go about doing this is we find a group of whales, we put some of those satellite link tags on, and then shortly before the trial begins, we also try and put a digital acoustic tag into the group as well on another animal. And then we conduct what are referred to as controlled exposure experiment trials. So we will either use a simulated sonar, which we have a sound source that we can simulate the Navy sonar, but preferably we actually bring a Navy destroyer in to play back sound to these whales in kind of real time, real life. And then we follow those tag whales to see how they respond. We get some data from the satellite link tags, and then eventually we recover the digital acoustic tags when they come off so that we can see the fine scale response. We had to work enormously hard to make those satellite link tags function effectively. This is Nicola Quick, who's part of our team. She's now at the University of Plymouth in the United Kingdom. So we had to tinker with the settings in the tags to make sure that we were getting data, especially during the period of time when the animals were exposed. We didn't want data gaps, and these tags tend to be very gappy because, as I said, the animals don't spend very much time at the surface, and we have to kind of get lucky if they come to the surface when there's an orbiting satellite overhead. So we tend to repeat the transmission of data over and over and over again to make sure that we actually capture the data. So, and just to show you how that works, these are two animals in the same group. It's the dive profiles of two animals. The black line is an animal wearing a digital acoustic tag, so where we have very fine scale measurements of depth. And the red dots are a satellite link tag in the same group diving synchronously, so the two lines should be exactly the same, and they are. Satellite tag only allows us to get measurements of depth every 5 minutes because that's as fine scale of data as we can actually pump up to the satellite. So, very proud to say that we've conducted experimental trials now with 9 Navy destroyers. This is probably the most complicated part of the entire experiment. Working with goose-beaked whales is difficult, but having a Navy destroyer with 300 sailors that's busy doing training exercises getting them to exactly where we need them to go and to do exactly what we need to do when we need them to do it is very complicated. And I'm very thankful to the officers and crew of these vessels that are all home ported for the most part, either in Norfolk or in Mayport, for taking time out of their really busy schedules to work with a bunch of pointy-headed scientists. So it's great. One of the things we really wanted to make sure was that we were never going to expose any of our tagged whales to sound levels that might cause them harm, right? We did not want a repeat of what happened in the Bahamas. So the sound levels produced by these sonars are very, very high, very intense levels of sound. So what we do is to position the Navy vessels typically 20 or 30 kilometers away from the tagged whales, and then we use real-time sound propagation modeling that was developed by the Navy to estimate the levels that the tagged whales should receive. And all you really need to know here is that in this case, our models suggested that a tagged whale would receive about 138 decibels, which is very audible, but not terribly loud. And the received level we measured on the tag was 138. So the models that the Navy has developed to understand how sound propagates in the water are very, very good. And that's not surprising because lives depend on those models. So just to give you a flavor of what this work is like, I'm not going to show you tons of data because I don't want to bore you, we have a manuscript in review now, but here's an example. So the sound source is up on the top of the plot, so the Navy vessel's up there, and then we have two animals in the same group, two of the same animals I showed you earlier, one wearing a digital acoustic tag, one wearing a satellite tag. We expose them to sound for about an hour, and what they do is to essentially move away quite rapidly from the source. This is in the map plan here, the XY. Again, consistent with the idea that they're perceiving this sound as they would a predator. Interestingly, though, so we have the two whales in the same group. The black line is the satellite tagged animal, so it's a longer period of time, and the green line is the digital acoustic tag, which we're getting sound. So in the red line shows the exposure, when the animals were exposed to the sonar. On the dive before, the animal was echolocating, the animal wearing the digital acoustic tag was echolocating, it was foraging. It's exposed to the surface on actually the end of its first bounce dive when we would expect it to make a whole series of other relatively shallow dives, but it doesn't. Instead, what it does, it goes silent, and it makes a kind of very long, very deep dive, and the bottom part there just shows you the net square displacement, the distance the animal moves away. So the animals go silent, and they move away from the source. Again, consistent with the idea that they're perceiving the source as a predator. This just blows it up a little bit. This is a dive-by-dive analysis, so you can see that first dive in red, that's the foraging dive. The animal comes to the surface, makes a shallow dive. We're expecting another shallow dive, and then the animal changes its behavior. And we think that this, although the animal's not at risk here because it's not responding in a very extreme way, but you might imagine that in a case where an animal was exposed to a very loud stimulus, suggesting that the predator was very close, an animal might make physiological decisions to either extend a very long dive or not come to the surface that could put that animal in physiological harm. So everything that we've observed so far, and we have done a lot of these playbacks. In addition to the 9 Navy vessels, we've done a lot with the simulated sound source. And everything that we have done so far is consistent with the idea that the animals are responding to these tactical sonars as if they were a predator. These animals basically use an anti-predator response that is based on crypsis. They try and hide. So they go silent, they move away. Interesting, one of the things we see is that the intensity of that response often increases at the end of the playback. Also consistent with the way that killer whales hunt mammals. So killer whales that hunt other mammals typically are silent when they're hunting. So they might be chatting with each other before they start a hunt, but when they start to hunt, they go silent because the things that they're trying to hunt, in this case beaked whales, can hear them, so they go silent. So imagine a beaked whale, you hear a sonar that you perceive as a predator, you go quiet, you move away, you're still able to determine where that predator is because you can hear it, and then it goes silent. When it goes silent, that means it's hunting, And that means you really have to move away rapidly. So that's how we do it. And I think the scale at which being able to do this is extraordinary. Nobody's ever put this many tags on these animals. We're very fortunate, as I said, to have a great team, a great study site that is in striking distance from shore, and the cooperation of the Navy. This is me in the pulpit of our tagging boat 40 miles off Cape Hatteras. And again, remember what I said, like you come across a group of beaked whales, they dive. If it's a foraging dive, you've got an hour to sit there and wait. So we talk about what we're going to eat for lunch. We have to be scanning the entire time because you never know when they might come up. These are some of the questions that we have puzzled over while we're sitting out there waiting for beaked whales to surface. First question, are they really mammals, or are they here from a different planet and we just think they're mammals because they're so different? I've studied whales my entire career. I've never studied anything that is so strange, so wonderful, so different from any other mammal. And again, just imagine what it's like down there, 1,500 meters. That's your safe place. That's where you go to get lunch. And it's a remarkable thing. We haven't solved that first one. The two other questions we do puzzle about for a lot is like, how, as mammals, can they stay submerged for such long periods of time? How do they do that from a physiological perspective? And then also, how do they organize their lives, their social lives? We know that they are pretty intensely social because we see animals in groups all the time. As I'll show you, the males have tusks and they fight with each other. How does that all work? So let's take the first one. We were interested in knowing what the aerobic diving capacity of these animals was. And we were interested in that just from first principles because they're such deep divers. But we also wanted to know how to interpret some of the long dives we saw as a response to the Navy sonar. So Nick Quick led this paper that we published a few years ago in which we tried to estimate how long an animal could stay submerged and with the oxygen supplies it took on board with it. So we used a technique known as a behavioral aerobic dive limit, and this is some of the data from Nick's paper. Again, very consistent with the early work that Jeannie had done. The average length of a foraging dive is about an hour, and only about 5% of dives exceed 78 minutes. And so the rule of thumb we use in the diving physiology world is that animals typically don't spend very much time exceeding their aerobic dive limit. So we ballparked the idea that animals probably have enough oxygen on board to stay submerged for 78 minutes. Any submergence below that, the animals have to start undergoing anaerobic metabolism. And so if you're a runner and you go for a run and you go for a very, very long run and your muscles get tired, that's because you're building up lactic acid. That's anaerobic metabolism. And you have to pay a price by resting after you run to be able to clear that stuff. But beaked whales don't read that literature. So for most dives, for every extra minute the animal stays submerged, they have to spend almost a minute in that subsequent into-dive interval at the surface doing those bounce dives, which we think might be kind of recovery dives. It's like you're walking after your run. But when we looked at those really, really long dives, the 5% beyond the 78 minutes, there was no relationship, which is weird. Because those dives should be the most taxing and should require the animals to rest. Every other diving mammal does that, and these guys don't. So we published this work, and we kind of raised some questions. We still don't really understand how they do it. But in that, we also published the documentation of one of our whales that stays submerged for 3 hours and 42 minutes, which is the longest dive that any mammal has ever been documented to make. We also have dives now on our tags that go to 3,500 meters. That's a 7-kilometer round trip. And again, how does a mammal stay submerged for 3 hours and 42 minutes? When humans do submergence contests to see who can stay submerged the longest, they typically go in a pool and sit at the bottom of the pool. These animals are actively swimming to go and get lunch or dinner. How does an animal stay submerged and do that for 3 hours and 42 minutes? We don't know. Of course, New York Times picked this up. They didn't care about the really cool stuff about physiology. They just liked the record. That's fair. So let's talk about social interactions. This is a male surfacing, and you can see at the distal tip of its lower jaw there are 2 teeth. These animals are suction feeders. They don't use teeth. Females don't have any erupted teeth at all. So they approach a prey, and as I said, they're feeding on stuff at the seafloor that is pretty slow moving, and they can just slurp them up using suction. The males have teeth that erupt, but they don't use the teeth for feeding. They use those teeth in fights with other males, in agonistic interactions with other males. Interestingly, this animal's got a couple of barnacles on its teeth. Those barnacles picked a very unusual place to settle, and I've often wondered whether these barnacles, like, how do they enjoy that regular cycle from 30 degrees Celsius water at the surface, down 1,500 meters to 4 degrees Celsius? I don't know, but it's remarkable they survived. So mostly thanks to the work that Will Trophy's done, I also say that we're able to identify animals in photographs, and Danielle Waples in my lab does all the photo identification. It's very time-consuming, very laborious work. She's great at it. We're able to actually document how groups change. And it turns out the beaked whales are much like bottlenose dolphins in that they have what we refer to as a fission-fusion society. Same thing that chimps and bonobos have, same things that we have. We're together here as a group. After my talk's finished, we'll go into a different group. Beaked whales do exactly the same thing. So here's some data from 2020. On the 9th of August, we saw tag 108, tag 71, tag 72, tag 76, and tag 106 together. All those animals had tags. We saw them 10 days later. 108 and 71 were still together, but there were 3 new animals, one of whom we tagged, 2 of we didn't. Then we saw them the next day. 108 is still together, 71 is still with them, 84 is still with them. They picked up another animal. So that's how these groups, that's what the dynamics of these groups are like. So the males are presumably moving from group to group, looking for females to provide them with reproductive opportunities, looking for females to mate with. And the females are worried about bringing up their kids, making sure their kids are safe, and eventually, when they become receptive, mating with the males. One of the really strange and odd things, out of all the strange, many odd things these animals do, that Will was able to document as part of his dissertation work, is that adult males in the same group display extraordinary synchrony. So here at the top plot is 2 animals, 87 and 89, tagged in the same group. They're both adult males. We can tell that from their pattern of scarring. And over this period of time, they display perfect synchrony. We expected them to be very asynchronous, and we hypothesized that adult males wouldn't stay in the same group for very long because they're essentially competing for females. You can compare that with 2 animals of different age sex classes, an adult male and a brown animal, we don't know if it's a female or juvenile, that don't display that kind of synchrony. So a couple of hypotheses here. Could males be working together to gain access to females? We know that bottlenose dolphins do this, and sometimes referred to as coalitions, in which males, bottlenose dolphins, form lifelong partnerships with each other to gain access to females. Certainly not permanent like this, because we don't see the same 2 males together all the time, but maybe for short periods of time they do. Or is it some kind of weird dominance interaction in which the animals are assessing each other, and part of that assessment is this weird synchrony? We don't know. We're also really interested in when socialization occurs. We think that for the animals to be able to make the extraordinary dives that they do, they basically have to shut off all non-essential organ system function. So we know that the classic diving response means that you slow your heart rate, you shut off your peripheral circulation, you maintain blood flow to your brain and to your heart because those are the 2 tissues that need it, and you basically do nothing else. So if the animals are doing that, they're certainly not socializing on those dives. They're not digesting, not doing a hope, they're not resting. They might sleep on the way down. When do they socialize? So presumably they're socializing in these bounce dives. But the weird thing with those bounce dives is the animals are silent when they're doing that. So if they're socializing, they're doing it in silence, which again is antithetical for a toothed whale. So we've been having students look at when animals are acquiring these new scars. You can see the scars there behind me. Those are scars of the agonistic interactions between or amongst males. And those last for years and years and years. And eventually animals become, old adult males become almost entirely white. We don't know how long these animals live, but we're trying to use the pattern of scar acquisition to give us some insight. And if I had to bet, I would bet that these are very, very, very long-lived animals. Females reproduce very infrequently. We do have examples in the whale world, bowhead whales in the Arctic that lived for more than 200 years. And if I had to put my money, I would say, if anything was going to challenge that, it would be this species. So maybe they live for 100, 200 years. We don't know. One of the things we have been doing is to obtain small samples of skin. This is an animal wearing a tag, and there's a biopsy dart on its way, fired from a crossbow, and we can get, with a tiny little surgical stainless steel biopsy tip, so we can get a small sample of skin and blubber, and from that sample of skin and blubber, we can genotype the animal, so we can determine its sex, we can look at its relationship to other animals, we can look at stress hormones. And when we combine this with the photographic record and the tag record, we get a really rich picture. So this animal we know is an adult male, you can see all the scars. But for those brand animals, often we don't know if they're male or female, if they're adults or juveniles. We would like to use kind of some epigenetic techniques to estimate age. We haven't done that yet, but that's kind of in our quiver of things we want to do. One of the things we've been able to do, leveraging some of the incredible resources we have at Duke, is to take some of those skin samples back to main campus in Durham and then grow cell lines out. So a lot of the questions we have about how these animals make their living, we can't answer because we can't bring them back into the laboratory and experiment with them or monitor them. But we can bring their cells back, and then we can grow their cells in the lab, and then we can expose those cells to hypoxia and anoxia, low levels of oxygen, to see what genes turn on, what genes turn off. And this is the work that's being led by Jason Somarelli of the Duke Cancer Institute. And it's I think it's a great example of how we can collaborate with folks who have the molecular tools to understand the physiology of these animals by bringing cell lines back from the field into the laboratory. So I'm going to leave you with this picture. This is the last one. This is a picture I took on the bow of the Barber, our research vessel, our tagging vessel. The animal's descending and it's going down in this beautiful azure Gulf Stream water down to forage. And I just want to kind of repeat this kind of crazy idea that surface is a dangerous place for beaked whales. They don't like to be at the surface. They're happy to anthropomorphize when they're down 1,500 meters foraging, when they're safe. And that's just a very difficult concept for us to grasp, but it helps to explain why we perceive these animals as such strange, enigmatic beasts. So as I said, everything I've done here is the result of work of an enormous number of people. I've tried to mention folks as I go through, but I just want to say thank you to the Navy who supports this work financially, and thanks to a whole bunch of other folks who have supported us in the field analytically. Thank Kim Urian, who is my muse, for helping make all my presentations better. So with that, I would be happy to take any questions. I'll leave you with this lovely picture from our colleagues at UNC Wilmington of a group of 5 adult males out in the Gulf Stream. So thank you.
All right. Thank you so much. Fascinating. Oh my gosh. If you're here in the room with me and you've got questions, want to know more, just wave at me. If you're on the first floor, I'll actually bring you a microphone. If you're up on the balconies, just wave down at me, and then we can call on you, and then you can yell at us, and we'll use the microphones to repeat the questions. And if you're watching online live on YouTube, you can type your questions into the chat. I'm keeping an eye on it, as well as the folks with the Office of Environmental Education. So we're seeing all of your questions on the internet as well, and we'll get those into the program. So if anybody's got one they want to get us started, the first question that did come in online for you is, have you ever seen killer whales hunting the goose-beaked whales?
We have not off Cape Hatteras, and that's because I think that killer whales are very scarce now in the Northwest Atlantic. They were largely eradicated back in the 40s and 50s. Amazing as it seems that military vessels used to target killer whales and shoot them because they were perceived as competitors for fisheries. So we have very few killer whales left. Pretty much every winter, though, we do see killer whales. We get reports of killer whales in Cape Hatteras for doing things like taking bluefin tuna off troll vessels. So you imagine like hooked up with a big bluefin tuna, killer whale comes along and takes your tuna. So even though that predators are scarce now, if these are very long-lived animals, they'll have the memory of what predation is like. It's like if I told you there was a mountain lion somewhere here in the museum wandering around. The chance that you're going to be taken by that mountain lion are pretty slim, but I bet you we close the doors, right? So.
Yeah, absolutely. Makes a lot of sense. I'm curious, the other deep diving whales that people know about, like if you ask most folks what's the deepest diving whale, and I have, playing games in the museum and stuff, they say it's the sperm whale. Are there insights that you can get from like a giant toothed whale? Because sperm whales are so much larger than these, must have very different physiology for diving. Are there insights that work across species like that?
Yeah, that's a great question. And they're mammals, and they're both mammals in the same clade. They're both cetaceans. So yeah, we can gain some insights. Sperm whales are not as extreme divers as beaked whales are. So sperm whales, we can make sense of what they're doing in a regular mammalian physiology that is kind of tweaked a little bit. Where beaked whales go is to extend that way, way out into very long duration, very deep dives. So what sperm whales do, we can make sense of. What the beaked whales do, in the extreme cases, we really can't make sense of yet. We're hoping that some of that molecular work will try and help explain what's happening. Maybe one of the things we've wondered about, for example, is with the goose-beaked whales, either they have a system in which they're not producing lactic acid like other mammals do when they undergo anaerobic metabolism, or maybe they have a new molecular pathway that allows them to process and do something with that lactic acid so it doesn't build up. Either one of those would be a completely novel physiological process, which would be fascinating from trying to understand the limits of mammalian physiology. Also, think about what we could learn from these animals in terms of human medicine. Imagine if we could train or somehow adapt our bodies so that we didn't have to breathe for an hour or so. We'd make things like surgery, all kinds of things. There'd be a lot of advantages to being able to do that if we understood how we could do it.
Do you have any guesses as to how these animals handle the pressures? I mean, especially this one whale that went down, you said 3,500 meters, 3.5 meters down?
No, I mean, it's an incredible amount of pressure. And so we know a couple of things. So the animals allow their bodies to conform to the pressure. So their ribs will kind of collapse. They don't use the air in their lungs at all as an oxygen store. All the oxygen they use is stored in their blood and their muscle. So we know those kinds of things. But if we were there, even if we had oxygen, we'd be crushed at those depths. I mean, those are the depths of the Titanic, right? We don't know how they do. We don't know how they deal with things like nitrogen narcosis. And so there are a whole bunch of physiological questions we really just don't understand. We would love to know the answer to that question. Think about how these animals find prey, right? They're using sonar, and the way that they produce sonar signals is to move air in from little pockets in their head across a membrane very rapidly. So the air in those little pockets is compressed enormously when they're at depth, but somehow they're still able to push that air back and forth to produce a coherent echolocation signal. We don't know how they do that.
Fascinating stuff. Okay, another one from the chat for you. Are juveniles able to dive as deep and as long as adults?
Great question. We don't know. We see very few, very small calves. I'll attribute this idea to Will Chofee. He wonders whether when females give birth, they might move out of the core area and go somewhere else for a period of time until their calves able to make dives. We don't, in some deep diving species like the pilot whales that we started studying, sometimes you'll see groups of calves at the surface with somebody babysitting them. We never see that with beaked whales. If you see a mom beaked whale with a calf, they both go down. We don't know whether the calf goes the entire way down or not, but likely, because otherwise how would they find each other again? So it's the ontogeny of that diving physiology, how that develops over especially the early period of life. Imagine how difficult that is for a baby beaked whale, right? You're born, You have to get to the surface to breathe, and then your mom goes on a 1,500-meter dive. I mean, holy smokes, that's crazy.
What do you and your colleagues make of that one world record dive? Like, yeah, the New York Times and popular media is certainly going to pick up on a world record like that, and it is impressive. But compared to the averages that are much shorter, much shallower dive, still deep, but not as impressive, say, what do you make of that?
Yeah, we do have another very, very long dive, and we wonder what the function of those dives were. Was the animal scared, really scared by something? Was there a particular prey source that the animal was going for? We really don't know, and we can't really find good reasons to stay submerged for that long, unless it was pursuing something that was a very valuable prey. And again, we know nothing about what they're eating down there. One of the things we're doing now, we have another project funded by the Office of Naval Research in which we are using environmental DNA to sample the waters where these animals dive, and we're trying to get down to 1,500 meters to sample the waters where their prey are. We can't go down and then catch things 1,500 meters deep. That would be a big rod and reel and a lot of line. But we can sample the water and get environmental DNA and then trying to understand what is down there the animals might be eating.
Thank you. Hi. I didn't catch the first part of your talk, so you might have answered the question, but you just were talking about how I didn't know about these whales, and a lot of people think that the sperm whales are the deepest diving ones. Does that make, would you like more people to know about your whales, or does that make a difference for you, for funding you might get, how popular they are, I guess?
Yeah, I think so. It's a great question. Sperm whales are iconic animals because of Moby Dick, right? Everybody has heard of Moby Dick or watched the movie or read the book in school, and we all have this idea of a sperm whale, this conception of a sperm whale. These are animals that most people are never going to see. There are no wonderful novels written about them yet, but maybe somebody will. That would be awesome. I would love for people to know more about them, and one of the reasons I'm here today, because I think it's so extraordinary that we share this planet with animals that do such strange things. One of the things I should have said at the beginning is this whole group of animals, this whole group of whales is so poorly known that we're still discovering new species. We're still discovering new species of whale. We are certain that there are a couple of species of beaked whale in the Pacific that remain to be described by scientists. And I think that's also extraordinary and wonderful. And maybe a 12-year-old girl somewhere is listening to this will be the person who describes that beaked whale in the middle of the Pacific 10 years from now or 20 years from now, and I think that would be wonderful. So the answer to your question is yes, absolutely. We should all know the wonderful variety and diversity and strangeness of life on this planet.
Fantastic, thank you.
Yeah.
So another question came in online for you. The question is, do you have an idea of what they're eating through necropsies? Can you give us some insight onto, besides the tags, how do we know what we do know about beaked whales?
So beaked whales strand from time to time, and we have a wonderful stranding network here in North Carolina. Vicki Thayer, who runs the stranding network in the central part of the coast, I think it's online. Hi, Vic. And she and Keith and their colleagues, and Annabelle and Wilmington and their colleagues who run the stranding program down there do great work to conduct very full postmortems or necropsies of these animals because we want to learn as much as we can about them. One component of those postmortems is to collect the stomach and then to look at the contents in that stomach later on to be able to make some insights into what they have been eating. Now remember that most of these animals that strand have been sick and have died. So we know that what's in their stomach is biased because animals are not very likely to have been feeding prior to stranding if they're sick. We also know that certain things tend to collect in their stomachs. So what we find in the stomachs of those stranded whales typically are squid beaks. So the hard chitinous beaks that squids have. So we can make some inferences into what the squid beaks, what species they're from, how big they are. But we think it's a pretty biased viewpoint. And we really don't have a good understanding of what it is that the animals are eating. We think that because the animals are so focused on the bottom, and we know from some wonderful exploration work done by NOAA with cameras that are on submersibles, there's a lot of stuff down there, big fish called like rat tails, deep water squid, and that's what we imagine that they're feeding on, but we don't have a good basis for making predictions of what species in specifically they're eating off Cape Hatteras.
Next one that came in, some of our viewers are postulating that the mother's long dives without the infants, could they be compared to birds leaving for long times to feed and finding their offspring in huge colonies?
So the way that birds do that, like they vocalize, right? They can actually, and same thing with seals who come back and leave their pups on colonies. The seal pup and the seal mom know the vocalization of each other so they can recognize each other. Tricky thing for beaked whales is they're clicking, they're making echolocation clicks, but they only do that when they get to about 500 meters. And in the top 500 meters of the water column, the animals are silent. So if a mom is going to leave its kid at the surface, how does it find again when they're not vocalizing? They don't want to vocalize because that would be making their presence known to potential predators. Maybe the kids can go down to 500 meters and stay there and wait, and then they can hear their mom coming back up. That's a possibility. But how all that works is still a mystery to us. But like I said, it's a great question.
And last one, does commercial fishing impact beaked whales? What is conservation status for this animal?
Another great question. So accidental mortality by catch in commercial fisheries is the biggest threat generally to cetaceans, whales, dolphins, and porpoises. We estimate that 300,000 whales, dolphins, and porpoises are killed each year in the globe's fisheries. One of the very few groups of animals that are not impacted by commercial fishing are beaked whales. So in general, we think that the biggest threat to their conservation is exposure to sound like this. And we worry in places where sonar is used repeatedly, like on Navy ranges, or where other loud sonars are used, that might drive these animals off preferred habitat. But in general, their conservation status is good. We're not concerned about them the same way we're concerned about North Atlantic right whales, and they do not get taken in commercial fisheries. And they were never hunted because they would be too difficult to hunt because they stay submerged for so long.
Well, everybody, let's give Dr. Andy Reid one more round of applause. Excellent job.
Thank you.
Fun stuff, fascinating. I bet you're glad you were here and glad you're tuned in because that was a lot of fun. That was a lot of really cool and interesting science and insights. And guess what? We do this every Wednesday now here in the SCCU Daily Planet Theater and on the museum's YouTube channel. So you can come back again next Wednesday at noon for the next Lunchtime Discovery Series. Meet somebody else cool and interesting doing fascinating work and gain more new insights about science and nature. So don't miss out. Make sure that you check out naturalsciences.org, the museum's website, to see upcoming programs and events. You can also go to the website for the Office of Environmental Education within DEQ. Their website is eenorthcarolina.org. There you can get contact information, find social media feeds, all the stuff that you want to do to stay connected with what's happening here in North Carolina. So thanks for coming out today, everybody. Again, thanks to Dr. Reid for joining us. Thanks to the museum's digital media team for all their hard work getting the program out. And we'll see you all again next time. Bye, everybody.